Welcome, my students. Today we will investigate the effect of different concentration of sodium chloride solution on the plant. This plant, or the potato plant, have been soaked in a methylene blue. Okay, as you know, it is one of the experiments that is applied on the osmosis. What is the osmosis? Who can say what is the osmosis? Osmosis is
diffusion of water from high concentration to a low concentration.
We cannot say the high concentration, because concentration of what?
Oh.
It is the diffusion of
The diffusion of
water
through different levels of concentration.
Actually, the same thing. It is the movement of water. Okay, you can say the diffusion of water. We can say the net movement of water particles from a region of their higher water potential to a region of their lower water potential. Okay, via partially permeable or selectively permeable. In the AB, we like selectively permeable because it is much more accurate. Okay, so first of all, we have a 20 centimeter of 10% sodium chloride solution, and here in they labeled W, it's a distilled water, 200 centimeter cubed, and here we are referring to the record. You will write your recordings here. Here we have two syringes. One will be used for the sodium chloride solely. You should not mix the syringe, and you have to rinse it after every single, okay, yes, exactly, in order to avoid pollution that may affect your results, because it is a source of error. Okay, so this one will be used for the distilled water. The potato was being placed or soaked in a methylene blue, which is a dye that is being taken by the plant cells. Okay, actually, before you came to the lab, we rinse the excess amount of the methylene blue, okay, in order to have a proper way of handling the experiment. Okay, so first of all, we have to prepare 5 different concentrations of the sodium chloride solution. How can we do this? First of all, we should use beakers, but actually I'm gonna use in those small cups. 1, 2, 3, 4, 5. So I have different, okay, 5 concentration. The first one, I will label it 10%, because the sodium chloride solution is already, okay, diluted to the level of 10%. Okay, so I'm going to write here 10%. So I'm going to I will take 20 centimeter. Here is 10 centimeter cubed, so you will take it 2 times exactly. So here 10, another 10, okay, and I will add 0 distilled water. So this concentration is 10, okay, exactly. Now I'm going to go to the second process. I'm now under the procedure of serial dilution. First, I have to rinse it, exactly. In order to avoid the contamination. Not only this, and I have to place it back again, okay, because I will use it for the solution. I will use a stirrer, okay. Shall I use it here?
No.
Yeah, exactly. Why? Yeah, I don't need it because actually I didn't add anything to it. Okay, afterward I have to rinse it. Okay, you can clean it or no need for that. So afterwards I'm gonna go for the second. I will take 2 centimeters of the 10% sodium chloride solution, only 2 centimeters cubed. Yeah, and here I'll add it. Okay, then I have to clean it. Okay, not to forget. Sorry. Yeah, I rinse it. I will return it back. Then I will take 18 centimeter cubed of distilled water. Okay. Sorry, it's not properly taken, all of it. So here is 10. Then I will take 8. Okay. So in the second cup here, I'm bringing it back again. Shall I need to clean it or no? Because it is actually a distilled water. I never mix it. Yeah. So here I have 10%. I took 2%. I took 2 centimeter cube of 10% sodium chloride concentrated solution, and I added to a cup, and I got 18 centimeter cube of distilled water. So what is the percentage of dilution here? It will be 1%. 1%. Exactly. I will write it down. 1%. 1%. Okay. Afterward, yes, here we have it. Here is 10. I will just make it a little bit bigger in order to be obvious. Here is 10. Okay. Yes. Here is 10. Here is 1. Okay. Then I will go for, if I took 2 centimeter cubed here and added to them 18 centimeter cube of the distilled water, what will be the serial dilution in this case? Yeah, if I took 2 centimeter of 1% sodium chloride, it will be 0.1. Yeah, I will write it first. It will be 0.1%. Exactly. Yeah, 0.1. So the procedure as follows. I will take here 2 centimeter. I will add them here. I will rinse it. I have here a fresh water. I can use it as here is a tap water. I can use it for a fast, cleaning. And I bring it back again. And I will take here 18 centimeter cubed. Here is 10. Additionally, 8. So I have here 20 centimeter of 0.1% sodium chloride concentrated solution. Afterwards, if I took 2 centimeter cubed of 0.1% sodium chloride, and added to it 18 centimeter of the distilled water, what will be the serial dilution here? 0.01, exactly. So it will be 0.01, yeah, 0.01, exactly. So here is the cup. I will take from 0.1, just 2 centimeter, put them here. I will clean it, not to forget. Okay, back again under the salt. Take from here 18 centimeter cubed. Sorry. Here is 10. Here is another 8. Okay. Okay. I forgot to do something. You didn't tell me. I have to stir here.
Yeah.
Okay. And after stirring, what shall I do? Rinse. Stir the second. And afterward I have to rinse. Okay, I'm dry in order not to have any even droplets of water that may affect the percentage of the concentration. Here I stir it and I will rinse it. Okay, dry it in order to be pretty sure that there is no additional. Yes. Now we have 10, 1, 0.1, 0.01. If I took 2 centimeter cube of 0.1% of sodium chloride concentrated solution and added to it 18 centimeter cube of distilled water, what will be the serial dilution here? What would be? 0.001, exactly. So it will be 0.001. Yeah. Yes, okay. So here we go. I'll just make it a little bit, okay, obvious. Okay. Perfect. Afterwards, I'll make it here. I don't need this anymore. I need it in a couple of seconds. Okay. So 2 centimeter of 0.01. I will. Yeah. Yes. Then I will take 18 cubic centimeter. Here is 10. Here is 8. Okay, I'm back again. Now, I have now 5 different concentrations. 5 different concentrations. Okay, so in front of me here I have 6 test tubes, but I made 5. So what did the 6 test tube will be used for?
Control.
Exactly. It will be only having distilled water without any sodium chloride. Why? It will be used as a control. What is the control using for? For comparison. For comparison, yes. So I will take 10 centimeter, okay, cube of each, okay, concentrated solution. From the distilled water, I will do the same one. Here we have the distilled water, and I label it with W, okay. Now, before adding the sodium chloride concentrated or different concentrated solutions, I would like to label them with their concentrations. Okay, so the first test tube will be 10, exactly, 10%. Okay, okay. This is 10. Next will be 1. Afterwards will be 0.1. Afterwards there will be 0.01. 0.01. Eventually we will have 0.001. I arranged them according to their concentrations, okay, quantitative, okay, concentration, yeah. So we have 0.001, the last one. So now we're gonna go for, okay, taking a 10 centimeter from each concentrated, okay, solution. I don't need this anymore, okay. Now I will use the same syringe, but I have to rinse it after every single, okay, use in order to avoid the contamination for, okay, yeah, exactly. So, are we there? Saleh, are we there? Okay. Please focus here. Yeah, so here we have 10, okay, it's not perfectly 10. Now it is, okay. So I rinse it in fresh water or tap water, okay. I will clean it, okay, to avoid any, okay, contamination. Then I'm gonna go for the second concentration, taking 10, okay, sorry, place it here. Now clean it, okay, different tissue, don't worry, to perfectly, okay, sure that there is no mixing. Then I'm gonna go for the third concentration, which is 0.1. Here we have 0.1. Then clean it, okay, dry it, 0.01. 10 centimeter cubed, clean it to avoid contamination, dry it. Then the last which is 0.001, 10 centimeter cubed, okay? And I will not use it, but I will do this, okay, to be sure. Yes, afterwards, okay, I'm gonna go for the next step. The next step, I'm gonna remove the potato from the dish labeled P, okay. Here are the potato that has been soaked in the methylene blue, okay. I will use a white tile, okay. Carefully, again, carefully, okay. Using the scalpel, okay. I will use the ruler in order to cut precisely 1 centimeter, okay, length and 0.75, okay, centimeter width, okay. The width is already maintained. We cut it 0.75, so I'm gonna go for the length. Perfectly, 1, yeah. I need to cut 6 pieces, okay. Then another, second, I think third, yeah, I need to do this one more time. I have to be sure. Yeah. Yes. So now I have 3 pieces, okay, equally cut to a length of, yes, exactly 1 centimeter. Now I have 3. I need 4 now. I have additionally 4. I need 4 more. Yes, okay. So here it is 1, and here it is another 1. Okay. So there is a waste, okay. I can use it at any time, okay. Afterwards, I don't need those. I can return this back, by the way, okay. Those, not need. Even those. Now I'll take each piece and I will dip it into one of those test tubes. Okay, I have 6. 1, 2, 3, 4, 5, 6. Okay. So I have to use the stopwatch. Okay, 10 minutes. Okay, and afterwards I'll continue the experiment. Okay. Use the stopwatch. Okay, please. Yeah. Thank you. Yes, it's now on. Now, I will make use of those couple of minutes, the 10 minutes, in order to interview the students and let them work. What are you doing now?
So what we're doing right now is we're making a cereal solution.
Cereal, okay, dilution?
Dilution, yes. Basically, this is 10% NaCl.
Okay.
And we're trying to find out how osmosis is affected by the percentage of solution for NaCl.
Okay, you are writing the concentration on a paper?
This is the first one. This is for before. Yeah.
Okay.
Yeah, but this is 10%. This is 1%.
Okay.
This is going to be 0.1%. This is going to be 0.01%.
That's perfect.
Yes.
Okay. Carry on. Okay, Abdul Majid, what are you doing here?
So I would basically take the ruler
Yes.
and the potato
Okay.
slices, and I would cut it exactly at 1 centimeter.
1 centimeter.
So it would be
The length
right
is 1
over-
centimeter?
Yes.
And the width?
0.7.
Okay, 0.75, yeah.
So you would cut it like this.
Yeah. How many pieces you will cut?
We need 6.
Okay, that's great. That's super. And you will place each one of them where?
After the solution, we put it over here.
Yeah.
We'll take some of the potato and put it.
So you have here how many concentrations?
We have 6.
5.
I mean 5.
Yeah, and I think the water will be used. Yeah. Why label the test tube here as W?
Because we need a control which is 100% undilated.
Yeah. So it will be distilled water.
Yes.
Okay. How many centimeter you will place in each test tube?
10 in each one.
10 centimeter cube. Okay. For how long you will keep it?
You keep it for 10 minutes.
Exactly. Thank you. Thank you very much. Carry on.
Welcome.
Thank you, Ibrahim. Okay. Now we have 10 minutes passed. we will take the recordings. First, we have the distilled water. The distilled water, its sodium chloride concentration is?
0.
Exactly, 0. Okay. I will arrange them from the lowest concentration to the highest concentration. So the first one is distilled water, which is exact same, the distilled water. The concentration of sodium chloride here, 0. Afterward, we have after we did the serial dilution, we have 0.001, and this should be here. Okay? Okay? And this 10 is supposed to be the last one. After that, we're supposed to have 0.01. Okay? Then we have to have 0.1, then 1, then 10. Again, let's say that we have to arrange them according to their concentration. The first concentration is 0. There's nothing, no sodium chloride. Afterward, we have 0.001, then 0.01, then 0.1, 1, and 10. Yes. Afterwards, I will take the potato, okay, bees from inside. Actually, it's better, because a little bit, according to the instructions, I will pour the content into the, okay, beaker labeled R, okay, here. It's not yet, yes, now. Back again. And I will use it against a white, okay, cardboard paper in order to have a better, okay, contrast. I'm looking here for the intensity of the color, okay. This is a distilled water. What about the color of the, okay, if I give you this question, what is the color of the piece of potato? Okay, from a scale of, let's say, plus, double plus, triple, okay, 4, 5. What is the color? Have a look.
Plus.
What is the color intensity here?
Plus.
Plus, okay. So in order to perfectly record your results, you need to make a table in which the first heading you have to have the percentage of concentration of the sodium chloride. The first one is a distilled water, which is, okay, yeah, labeled W, and it is 0, okay, percent. If you, afterwards, 0.001, 0.01, 0.1, 1, and 10, okay? And here you have the second heading. It's the color intensity, okay? So the first one is, okay, plus, okay. Any one of you has any explanation for this? Yeah.
Can I guess?
Yes.
Is it that there's no like salt atom which doesn't break down?
So the water move,
It moves like slower.
the net movement of water is
Slower.
actually, anyone has any objections about this? The net movement of water inside is higher, because in this case it's a distilled water. The distilled water is high protonic solution. So the water will be moving from a region of their high water potential, in this case the distilled water, to a region of their lower water potential, which is inside the potato. So the color intensity of the potato will be less, because more water gets in. So the methylene blue dye inside the plant tissue is diluted. Exactly. Yes. So afterward, I will discard it in the waste. Then I'll clean it for my own, okay, purposes, in order to be sure that there is no, okay, water that will affect our results. Then I will take the second concentration, which is 0.001, okay. Yes, if I have a look here, the color intensity is
Plus.
okay, comparison to the previous one. If you compare it to the previous one, it's
Slightly.
slightly intense, okay. I give it double, okay, plus, yeah. Anyone knows why? Sorry? Anyone knows why? Is lower than in comparison to the distilled water. So there will be a net movement of water from the solution to inside the plant, but in comparison to the previous one, it is less. So the amount of water inside is less. So the color intensity inside the plant will be higher, will be more. Yes, then I will discard it. Same procedure. Then I will go for the second concentration, which is 0.01. Okay. Yes. What about the color intensity here? If you have a look here, it is.
Intenser.
It's more. Yes. Yes. Exactly. Exactly. So I will give it here triple plus according to the scale we are using. Then I will discard it in the waste. Okay. Clean it. I shouldn't actually. It is not a must, but just for better results. Here is the R. I will go for 0.1. Okay. It's not yet out. Yes. If you have a look here, it will be. Yeah, have a look here.
Looks way more intense.
It is really, really, yes, more intense. Okay.
This is the 1%?
There is, yeah, 0.1, not yet 1. Yeah, very, very, okay, intense in comparison to the rest. So I will give it 4 plus, then I will discard it, go for 1% of concentration. Even you can notice the color, okay, the dye go out, okay, reason, because the water from inside the plant is going out from a region of their higher water potential, which is inside the plant, to a region of their lower water potential, which is the solution, because this solution is now hypertonic to the plant cells. You got it? Yes. So if I look at it, it will be more.
Dark, it's like dark blue.
Exactly. So I'll give more color. Okay. I'll discard it. Yes. Then I'll clean it. The last one is 10%. Okay. So if you look here for this one, you're gonna find a lot of, yes, water go out, and even the color intensity against the white tile is really, really dark blue. Okay, as you can see here, the color intensity is really, okay, high. So this is the highest color intensity. Okay, and here will be the table that you are using. Then you can, okay, use the second paper for making a plotting the graph, in which you will, yes, use the previous recorded data, okay, that you obtained. Okay. So what we conclude from this experiment?
Higher salt in a solution will cause it to give higher intense of light.
Again? Yes, please. Hamad? Come. Yes. What we conclude from this?
Higher salt in the solution allowed it to create more intensity of color in the potatoes.
How come? How can you explain this?
Breakdowns, because water moves from higher to lower solution, so it just moves
Water move from high
to low.
High what?
Yeah.
High what?
High water pressure, high water solution.
High water solution or water potential?
Sorry, water potential. Yeah, from high water potential to lower high water potential. So
Okay,
as you can
so
see,
in this case, when we have a concentrated solution, which is the last one in this case, so outside of using this, for example, outside there is a hypertension or hypertonic solution. Hypertonic solution means
hypertonic solution from low water potential with high solution.
Exactly. So more solute and less water.
Yes.
So in this case, the water inside the plant is hypotonic in comparison to the outside.
Yes.
So it has higher water potential outside or inside?
High water potential inside.
Exactly, the inside the plant. So the water will be moving from inside the plant to
Outside of that.
exactly. So the color intensity will be there.
Darker.
Exactly. Thank you very much. Thank you. Have a nice day. Assalamu alaikum.